that he's back in Catalonia, where he's the group leader, as she said, in Hospital del Mar, Medical Institute, IMIM, where we have also our member, Ana Vigas, also there, and the leader of the Cancer Stem Cells and Metastasis Dynamic Lab. And it's a great pleasure to welcome you. Thank you.
Thanks, Manal, for the great presentation. Yeah, and time flies indeed. 4 years, I can believe it, but it's already done. So it's a great pleasure to be here today. It's my first time at this institute. I heard about it, but I never been here before, and I've seen that it's fantastic. So I'm very happy to be here. Also, I have many friends and colleagues here, so it's great to come and visit you. So I see the bar here. Is there a way to remove it? Yeah. If not, we'll deal with it. No problem. So today, I mean, we focus on breast cancer metastasis and research in the lab. And today I'm going to talk the last study and the first one of the lab that is about breast cancer immunotherapy. We are interested and we study in the lab, we focus in studying the phenotypic heterogeneity of the tumors. And we do it from trying to understand from the development to the metastasis. So how different phenotypes evolve during the progression of the tumor, how there are phenotypes like cancer stem cells, for example, that can lead this progression from the primary tumor and are critical and essential to initiate the tumor growth in the metastatic site. And for that, we also try to understand how these molecular mechanisms become aberrant in the cancer cells that are inherited from the normal mammary stem cells. So phenotypic heterogeneity, when we talk about intertumoral heterogeneity, we are normally used to talk about genomic heterogeneity, but in this case, phenotypic heterogeneity is, can I remove that or microphone? Well, I'll continue. So the phenotypic heterogeneity is, you can imagine the tumors are not heterogeneous. There are different population of cells with different behaviors and functions. So we can find cells like stem cells or more mesenchymal-like cells and cells that have different behaviors within a tumor. And this is elicited by extrinsic factor or intrinsic factors. And this heterogeneity can evolve during the time, the localization, and also change based on therapies. So this is a big challenge to characterize in tumors and has been, I cannot pass my slides. Okay, don't worry about the bar, we'll deal with it. So recently, this is again, the hallmarks of cancer, updated very recently by Hanahan in Cancer Discovery. And now finally they include, or they consider now the phenotypic plasticity as one of the hallmarks of cancer. We already knew that this is actually very important, but also the new technologies like single cell technologies allows us to study more in depth the cell populations and how they evolve in the tumors. So in particular, when we talk about phenotypic heterogeneity, we care a lot about what we call cancer stem cells. These are a population of cells that inside the tumor, they have a better ability or a better ability to initiate the tumor growth. These are aggressive cells, poorly differentiated cells. So these are populations that are less differentiated than the rest of the population, that they can derive other cell types, and they are normally resistant to different therapies, chemotherapies, radiotherapies. But now what we want to understand is what about immunotherapy resistance? Because it's actually not very well known how cancer stem cells are implicated in the immunotherapy resistance. And this is sort of a kind of a spoiler now of my talk, but we are particularly interested in how these cells are visible and how they express the antigen presentation machinery, so presenting antigens to the immune system in order to respond to immunotherapy. What is known so far about cancer stem cells and immunity is more related that they can express immunosuppressive ligands, and this is recent publications about that. Like they express more PD-L1, but actually PD-L1 is the target of immunotherapy, though. So based on that, they would be a target for immunotherapy, not the reason of resistance. And also they can induce an immunosuppressive tumor microenvironment. So we are interested on this aspect of cancer stem cells because during my postdoc, Princeton University, we found that normal stem cells in the mammary gland and cancer stem cells have a deficient response of interferon. And this is because they downregulate this factor that is called LCOR that we found to be important for the mammary gland differentiation, but also for the response to interferon from the microenvironment, specifically released from the macrophages in the mammary gland. So we found that LCOR is actually expressed in the luminal lineage of the mammary gland. So it's involved in the differentiation of the luminal cells. You can see how the luminal cells here express LCOR. This is in the normal human tissue, and the basal layer do not express LCOR. If we do mammary regeneration assays, so we put the cells and we try to exploit their capacity to act as a stem cells in a regeneration assay, we can see how LCOR is actually inducing differentiation and avoids the branching, avoids the regeneration of the mammary gland, and induce differentiation with expression of luminal markers. If we move this to cancer, we can see how LCOR reduce the expression of typical stem cell factors, embryonic stem cell factors that are also involved in breast cancer, how the breast cancer stem cells population by LCOR expression are reduced if we do knockdown, are increased, and specifically this a strong, very strong correlation and association with the interferon response. So one of the take-home messages of this study is that how we have a population of cancer stem cells poorly differentiated that do not respond well to interferon, and a differentiated and then LCOR-induced differentiation, and these differentiated tumor cells can respond very high to interferon. So that means we wanted to study how this can have an influence in the interaction with the immune system, because interferon has so many pleiotropic effects. As I told you, the intrinsic effects are differentiation, growth inhibition, but it has also a lot of extrinsic effects related to the antigen presentation and the engaging of the immune system. Moreover, interferon also is involved in the suppression of tumor progression, and collecting the literature, we can see how there are different ways to avoid interferon, and how with tumor suppression, there is tumors, they have the trend and they need to avoid interferon signals. So, so we want to study how interferon influence the antigen presentation machinery. Interferon, as you know, is an antiviral, is evolutionary conserved as an antiviral response in epithelial cells. And this is because it activates the antigen presentation machinery. So from the immunoproteasome, transporters, and the MHC molecules. So basically, the immunoproteasome can cleave proteins and generate peptides that can be antigens. These antigens need to be transported by TAP complexes and tapacin into the endoplasmic reticulum, where it's going to be loaded to the MHC molecules and finally presented. So we are interested in how the entire pathway is regulated, for all these components, how are regulated, because, for example, the transporters is one of the main bottlenecks of for the presentation, for the final presentation of these antigens. Most of the antigens are not actually presented because there is a bottleneck here. So if this is happening, then you can have the recognition of the T cells. And now we think that this is extremely important for the tumor immunology. Indeed, when we look at the clinical data from immune checkpoint blockade therapy resistance, so this is the modern immunotherapy, you can see how more than 50% of the cases of resistance are due to defects in the antigen presentation or interferon pathways. And these are other mechanisms related to immunosuppression. So here, one of the main goals of immunotherapy is reduce the immunosuppression of the tumors. So we have many drugs here, anti-PD-L1, anti-CTLA-4, anti-LAG3, that are designed to avoid immunosuppression of the tumor cells on the immune system. But then we have all these cases that are related to the immune visibilities, how tumor cells are visible or not to the immune system. And here we don't have specific drugs to actually manipulate this aspect. And as you can see, it's one of the main reasons of immunotherapy resistance. So going a little bit backwards and introducing immunotherapy. As you may know, immunotherapy works very well in certain tumor types like melanoma or lung cancer with very robust responses. But in breast cancer, it's actually not working very well. It is approved in triple-negative breast cancer, which is the only subtype that has certain responses to immunotherapy, but yet are modest responses. This is the first clinical trial approved in 2019 that show a moderate response in metastatic patients of triple-negative breast cancer. So we think that we have an opportunity now from the biology perspective, trying to understand why breast cancer and why triple-negative breast cancer are not as responsive as other tumor types to immunotherapy. And triple-negative breast cancer, of course, as you may know also, is one of the most aggressive cancer subtypes because it has a very high relapse, especially in the first years of diagnosis, so it's evolving quite fast. And there is a lack of efficient targeted therapies when there is relapse. And so the prognosis is poor for these patients. So, and immunotherapy is one of the candidates trying to increase the clinical benefit for this subtype of cancer. So with all that, we design a project where we try to understand the antigen presentation machinery regulation in triple-negative breast cancer and how phenotypic heterogeneity within the tumor can be important for the immunotherapy resistance. So for starters, we look at cancer stem cells. This is a triple-negative breast cancer patient-derived organoid. This is a light sheet imaging in 3D, where we have a reporter of cancer stem cells. This reporter is basically depending on the activity of SOX2 and OCT4. And we know that it can delimitate well what are cancer stem cells in the tumor. And as you can see, the green cells, they do not express any of the HLAs. So this is staining again pan-HLA. And the source6 negative cells, the cells that are not cancer stem cells, you can see how they stain well for HLA. So showing how in patients of triple-negative breast cancer, actually cancer stem cells are not expressing MHC molecules. And we did in many different cell lines of breast cancer that were annotated here. But in general, you can see how in different cell lines, the cancer stem cells in red, all of them have downregulated MHC molecules. B2M and also HLAs are always downregulated in the cancer stem cell populations of different triple-negative breast cancer cell lines. Also trying to understand if this is a conserved mechanism that comes from the stem cell biology. We also analyze this in mammary stem cells, and this is using public data from a paper from the Jeffrey Walls lab in the Salk Institute, where they isolated the different populations from fetal mammary stem cells, which are in the embryonic stage, and then the adult basal progenitor, luminal progenitors, and mature luminal cells. So here from left to right is increasing the differentiation. And as you can see how all the different components, not only MHCs, but also the transporters and the immunoproteasome molecules are increasing the expression when there is the differentiation of the cells and have very low expression in the stem cell phenotype. If we look this in cancer stem cells, so the entire antigen presentation pathway machinery molecules, again, also in triple-negative breast cancer cells, MDA-MB231 cells, all these components of the antigen of the APM are downregulated in these cells. And then going from patients. So this is data from 4 different patients from Hospital del Mar. So we isolated the cancer stem cells and the non-cancer stem cells. And you can see how in all of them, there is an important downregulation of all the components of the antigen presentation machinery in cancer stem cells from clinical samples. So how can we measure the entire pathway, the activity of the entire pathway? So we use this model, the OVA system, where we introduce a chicken of albumin in the cells. And this is a system that is very well used in immunology, very famous. So ovalbumin is going to be cleaved by the immunoproteasome and generate the different peptides. One is this yellow peptide that we know that is going to be generated that we call symphycal peptide, that there are commercial antibodies to specifically detect it. So by doing FATs, we can measure the entire activity of the pathway from the immunoproteasome, transport, and presentation. If we do that, we see how in cancer stem cell, in breast cancer cells, there is a population with high OVA presentation and a population with low OVA presentation. If we segregate the cells in using cancer stem cell markers typical of breast cancer, the CD24 low, CD44 high, which is this population here, you can see how the cells that express low levels of OVA, the cells that present low levels of OVA are segregated into the cancer stem cell population. Instead, the cells that present high levels of OVA in the surface, that have all this machinery very active, they are very reduced for the cancer stem cell population. So just by the presentation and the activity, we can distinguish cancer stem cell and non-cancer stem cell populations. Also, these low OVA presenting cells have more expression of stem cell transcription factors and less expression of LCOR, that protein that induces luminal differentiation. If we transplant this population, we separate by sort these populations, and we do limiting dilution assays in immunocompromised mice. So we wanted to measure the ability to growth of the cells that present different levels of OVA independently now of the immune system. You can see how the OVA low cells increase a lot the tumor initiation cell frequency, which is a typical capacity of cancer stem cells. So basically, this is telling us that we can separate a stem cell phenotype based on their ability to present antigens. So based on this, we did this assay in vitro where we use lymphocytes from a transgenic mouse model, that is the OT1 model, that express a specific TCR receptor to recognize the OVA peptide in the tumor cells. So these tumor cells have the OVA, and these ones have the receptor to detect and kill. So basically, with this assay, the cells that are not presenting the OVA won't be killed, right? So doing this assay, put in the co-culture, you can see how there is already a selective killing of the non-stem cell population, so the cells that present OVA, and the cancer stem cell population, when we put also immunotherapy, is the population that survive, meaning, again, according to the previous data, that cancer stem cells do not present the antigens, so they are not detected, and they survive. This is just a video to illustrate the same, how the non-cancer stem cell population are easily killed and not the cancer stem cells in 2D. This is a very similar assay, again, using the sources reported, again, with the co-culture and with the immunotherapy. The cells that survive are basically the cells that have the reporters of the cancer stem cells. And then we move to the preclinical assays, trying to understand the same, but in vivo instead of in vitro. So we did this assay where we grew mammary tumors in the mice, and then we started a periodic treatment of immunotherapy. So we had a control condition without immunotherapy, another condition with immunotherapy. You can see how these tumors initially responded to the treatment, and they stopped growing, but eventually they developed resistance. And we took these tumors and we did transcriptomic analysis. And the top down-regulated pathway, as sort of expected, but it's very nice to see it actually, the top down-regulated pathway was the antigen presentation machinery. So these resistant cells actually are cells that have the antigen presentation machinery downregulated. And among the upregulated pathway, we can find stem cell-like pathways or signatures. And these are also stem cell-like signatures that are relevant and has been published to be very relevant with aggressiveness of breast cancer and cancerous stem cells. And also in the IRT, the resistant tumors, you can see how they are associated with an enrichment of these signatures of these resistant tumors. And so again, if we do stem cell markers for this model, we can see how after the immunotherapy, there is a high enrichment of the cancer stem cell population. And again, these resistant cells, if we do the tumor initiation assay by limiting dilution, we can see how the tumor initiation cell frequency is increased robustly. But more importantly is where we take data from patients. You have to think that in breast cancer, till 2019, it was not approved. So there is limited data of assisted from the clinical assistant perspective, but we have some data from clinical trials. So we collaborated with Marlene Koch at the NKI Institute, that they had this clinical trial with metastatic triple-negative breast cancer, 53 patients. 9 of them were responders to anti-PD-1 therapy. 44 of them were not responders. So they had transcriptomic data, and then we analyzed the data, and we show how the non-responder patients are the ones associated, have enrichment of these cancer stem cell signatures here from the different studies. So this is showing how also these patients, the clinical patients that they do not respond well to the immunotherapy, they have a transcriptome that is enriched in these signatures typical of cancer stem cells. Then we also did a humanized system, trying to show also in vivo and preclinical assays that human cells can also do the same. We have a knock-in reporter of LCOR in these cells, so we can measure the levels of LCOR, and remember LCOR is this factor that induces differentiation in the cells. And then we crossed it with the sources reporter, this reporter of the cancer stem cells. In this humanized model, we used PBMCs. So you can see how the effect of the PBMC reduced tumor growth. The effect of PBMCs plus PD-L1 therapy stops pretty well the tumor growth. And with the treatment, you can see PBMCs and here the PD-L1 therapy. You can see how there is an enrichment of the cancer stem cell population reported by the sources reporter and the negativity of LCOR. You can see also here, these are different tumors, different mice, how the percentage of sources positive cells is increasing with the immune pressure, and LCOR is decreasing. And it's also very nice to see that this is also the same in patient samples. We had a limited number of samples. As I said, these are samples from clinical trials. I'm very limited to have pre and post treatment samples. So we got 4 samples from patients, but in all cases, we can see how this patient that developed resistant, in the resistant tumors, LCOR was decreased as it is decreased here in our preclinical data. And you can see here how these post treatment samples that are sample resistant, they basically shut down the LCOR expression. So far, and taking a breath here, I want to recap that within the tumor heterogeneity, the phenotypic heterogeneity, we have cancer stem cells that are more invisible to the immune system, and differentiated tumor cells that are the ones that express antigens or present antigens and are more visible to the immune system. So when we apply immunotherapy, these differentiated cells are eliminated, but the cancer stem cells are the ones that escape and lead immunotherapy resistance. And this is one of the main novelties or conceptual novelties of our study that was recently published. But how do they shut down APM? We want to understand, of course, the molecular mechanism of that. And I already introduced you a little bit LCOR. And so the response is LCOR. We have this cell line for a knock-in that reports with GFP the endogenous level of LCOR. You can see how in triple-negative breast cancer there is heterogeneous expression of LCOR. And so we can separate this population by flow cytometry with negative, low, medium, and high. And you can see how these populations, as more LCOR they have, more expression of all these APM molecules. This is also correlated with clinical data. If we stratify patients by expression of LCOR, you can see how the antigen presentation machinery signature, it is enriched in the patients with high LCOR expression. And if we do gain and loss of function studies, overexpressing LCOR or knocking down LCOR in triple negative breast cancer cells, we can shut down the antigen presentation machinery or we can turn it off. So this is basically the effect of this genetic modulation. So what is LCOR? I already introduced you a little bit, but I didn't talk a lot about the structure of the protein and what is described. So it was in 2003, it was described as a ligand-dependent co-repressor of nuclear receptors. For example, estrogen receptor. But here, what I'm showing you is in triple negative breast cancer. And triple negative breast cancer is ER negative, right? Or express minimal levels of ER. But one of the canonical functions of LCOR is to bind to ER and co-repress genes. And it does that through this motif, the LSKAL motif, because it interacts with nuclear receptors and not only ER, also progesterone, vitamin D, and others. But then what we found is that it has another domain that is called the HTH domain, that is a DNA interacting domain. And we did a conservation analysis of this domain in collaboration with Mar Alba at the IMIM. And so they found that these two domains, the nuclear receptor binding domain and the HTH, are very well conserved in vertebrates. But beyond vertebrates, only the HTH domain was conserved. And it's conserved in very simple organisms, in vertebrates, but also in prokaryotes. So it's a very ancestral domain that is evolutionary conserved in LCOR. And so what we did mutants for these two domains. We generated a mutant of the HTH domain, like in this DNA binding domain. And we generated a mutant with a change of the amino acids interacting with nuclear receptors. So this LSKAA mutant cannot interact with nuclear receptors. And we see that if we avoid interaction with nuclear receptor, nothing happens. So it acts similarly as LCOR, is increasing the expression of this machinery. If we delete the HTH domain, the DNA binding domain, then LCOR loses this function inducing the APM. But more striking was when we did the ChIP-seq analysis in MDA-MB2C1 cells. And these are the peaks represented across the entire genome of these cells when we overexpressed LCOR and we did the ChIP-seq. And we found specifically this larger peak of concentration of LCOR peaks in the genome in the short arm of the chromosome 6. And at the moment, we didn't know, but it was amazing to see that the entire antigen presentation machinery genes are evolutionary conserved, all in the short arm of the chromosome 6. This is called the MHC cluster. But also the rest of the proteins, the transporters and the PSMB proteins, the immunoproteasome proteins, are all clustered here. The only gene that is not clustered here of the entire antigen presentation machinery is the B2M, that is in another chromosome. LCOR also binds there. But the rest of the genes are evolutionary conserved here. So that was a very striking finding for us. And then we did a motif discovery analysis, and we found that the top motif enriched for the binding of LCOR was the interferon stimulated response element. This is the element that interferon regulatory transcription factors bind to mediate the responses of interferon in the cells. But LCOR can also bind to this motif. And so we built a reporter to measure the activity and how LCOR can induce transcription activity of this reporter with 6 tandem repeats and a mutated form. And you can see how LCOR and LSKAA can increase the reporter expression. So meaning that can increase the transcription of genes that has this motif. And also in the LCOR condition, we could see how it recruited the polymerase to indicating activation of transcription, which is a function that was never described for the gene. And then another very important finding of this study is that LCOR can do all this independently of interferon. As I told you in the beginning, interferon is what is described to modulate the antigen presentation machinery. But we found that LCOR is not only cooperating with interferon, but also can do the job independently of interferon. And so we crossed our knock-in of LCOR GFP with this reporter, the interferon response element reporter. And we can see how the more levels of LCOR, more activity of the reporter. And this is, can be shown here better. This is LCOR negative cells, medium and high. As we increase the LCOR levels, the reporter is increasing. If we put interferon, there is a synergic effect, of course. But then if we block the interferon signaling with an inhibitor of the JAK-STAT pathway or an inhibitor of the STING pathway, that are the two main pathways activating interferon signals in the cell, you can see how LCOR, high levels of LCOR in the blue, still can activate the reporter. So it's not depending in the presence of interferon. And moreover, if we do knockdown of LCOR and we treat with interferon, interferon cannot activate the activity of the reporter. So meaning that interferon to do all these effects require LCOR for the activation, at least for the gene that contain this domain. Then this is a similar concept, but by flow cytometry. So you can see here the control, the overexpression, the couples of control overexpression and control knockdown. You can see how LCOR can increase the levels of HLAs. If we put interferon, this increases a lot. However, with the knockdown, interferon cannot increase the HLA expression. And if we inhibit interferon, still LCOR can do the job. So this comparison are trying to highlight this independence of interferon. You can see how even the knockdown of LCOR, even if we treat the cells with interferon, cannot upregulate more the levels of HLAs more than the basal levels, and LCOR can still do the job when there is ruxolitinib, which is the inhibitor of the JAK-STAT pathway. So with that, of course, we need to prove that this can actually promote killing of these cells that present more antigens. So we co-culture in a similar assay as I showed you before, how, I'm losing the pointer, okay. How the LCOR overexpressing cells are going to be killed by the immune system co-culture. So you can see here in blue the condition of cells overexpressing LCOR with the OVA system. When we co-culture them, they are fully eliminated by the co-culture with the immune cells, T cells in this case. And if we eliminate, for example, B2M, so we avoid, B2M is this blue molecule here, so we avoid the final presentation of the antigen, we see that this elimination is dependent, of course, of antigen presentation. It's not another effect of growth reduction. So, so LCOR basically mediates this vulnerability to the immune system, and this is just a video to represent the same, how this LCOR cell is easily killed by the co-culture with the immune cells. In vivo, in the tumors, we can see how there is more immune infiltration and CD8 lymphocyte infiltration. This is tumors, different tumors measured by FACS. So the CD4 and CD8 populations are increased in these tumors. And if we take data from a cohort or from a data set, the Metabric data set that has more than 180 patients of triple negative breast cancer to have a large cohort data. And we do the convolution analysis of the immunophenotypes, how we can classify these patient tumors with immunophenotypes. The immunophenotype 4, which is the one that is supposed to be highly infiltrated with cytotoxic T cells, you can see how these tumors also express higher levels of LCOR, supporting our preclinical observations. So what we have now is an optimal configuration to respond to immunotherapy, because LCOR, if the cells that express LCOR, by indirect effects of interferon sensitivity, can upregulate moderately, not a lot, PD-L1, which is the brake that puts to the immune system, so immunosuppression, but it really increases, as I have shown you, the antigen presentation machinery. So with immunotherapy, with anti-PD-L1, we can basically release the brake and have a system where we can favor a lot the response to immunotherapy in breast cancer. Indeed, the LCOR levels are associated with response in these two different cohorts of clinical trials of breast cancer patients. So there is an association increase of LCOR expression previously to the treatment in these patients. Remember that it can also happen what I showed you before, that tumors can still be resistant if there are population of cells that actually take over after the therapy. So it's nice to see this prediction, but actually what we think that is more powerful of our system is to design a possible therapy. So the idea here would be when we have invisible cancer stem cells that express low levels of LCOR, so the idea would be to introduce them LCOR, do a molecular reconfiguration, and make these cells visible to the immune system. So we did this in preclinical assays. We can see we let the tumor grow to this size, and then we start the treatment with anti-PD-L1. You can see how the LCOR condition with anti-PD-L1 treatment in green, the tumor volume rapidly decrease, and these tumors, they don't grow never again. This is a waterfall plot of the same that is easier to see how the green cells, or the green condition, sorry, which is the anti-PD-L1 with LCOR, all of them have a complete response. So all the tumors completely disappear. Actually, 49 out of 50 tumors had a complete response. So all these tumors disappeared. 0 tumor disappeared in the control condition. We left 15 of these mice in the animal facility without treatment for more than a year, and all of them had a free tumor mammary gland. So all these mice were cured for the long term. We also can cure experimental metastasis conditions with LCOR and anti-PD-L1. You can see how the lung metastasis disappears, and there is an important reduction of the overall metastasis and survival of these mice with LCOR and anti-PD-L1. And so, of course, making this a therapy is very challenging. We are talking about the transcription factor. So what we thought, we had the idea, inspired a little bit about what was happening at the moment, we was the COVID and all these vaccines, the mRNA therapy, was try an mRNA therapy in our case. So for that, we use exosomes or extracellular vesicles. Indeed, we have a large percentage of exosomes here, 100 nanometers, and some extracellular vesicles that are characterized here in collaboration with Héctor Peinado at the CNIO. And we introduced LCOR mRNA in these cells to have the mRNA packed in the exosomes, or it's EVs. I have it here labeled as EVs. So you can see how the mRNA is indeed packed in these EVs, in the extracellular vesicles, when we measure the mRNA inside the vesicles. And so we can use the vesicle to treat cells in vitro. And we can see how this mRNA of LCOR that is fused with HA, we can see how HA is actually expressed. So it's the mRNA is translated, and we have HA protein that, of course, the cells wouldn't have unless there is translation, sorry. And also you can see how this HA that is fused with the LCOR is localized in the nucleus of the cells, as it is the normal localization of LCOR. So we also proved in vitro that this treatment can reduce the cancer stem cell population. It can induce the antigen presentation machinery, and also it can induce the vulnerability of the tumor cells to the immune attack in vitro. Then the next challenge, and I think the most important challenge, is if this is going to work in vivo. So we did a proof of concept of that, treating the mice with EVs retroorbitally. So this goes through this pulmonary circulation and goes to the lungs. And these mice had lung metastasis. And you can see how the administration of these extracellular vesicles with LCOR, we can also detect HA, the protein LCOR translated with HA in the tissue and with a high percentage of the cells of the tumors with this expression. And so we can do actually periodic treatments with immunotherapy later. So this is what we did, treatment with EVs of LCOR with anti-PD-1, and actually this worked. So we were able to cure also a good number of mice. You can see how the metastasis of this condition, the treatment of the extracellular vesicles with anti-PD-1 decreases metastasis, and also at least 50% of the mice had a very long-term survival, and the lung metastasis in most of the cases actually disappeared. So this is a summary of these findings of this study that I just presented. So basically, just to recap, when we have phenotypic heterogeneity, we have the pink cells and the blue cells. The pink cells are differentiated LCOR high cells that LCOR can induce the antigen presentation machinery. The blue cells are cancer stem cells without LCOR, and so they reduce the antigen presentation machinery. And these with immunotherapy are leading the acquired resistance to immunotherapy. These blue cells at the end are leading the acquired resistance. And then what we did is using this mRNA therapy. So basically we homogenize the tumor population and converting these blue cells in pink cells. So all of them having LCOR. And so when we apply immunotherapy, they respond very well. And then these mice at least were cured in preclinical settings. So going back to this idea, what we are showing here is that we are not only focusing or providing therapy for immunosuppression, but also for immune visibility, and how this is actually essential for the success of immunotherapy, at least in breast cancer. And so we are designing kind of a new mRNA therapy in the immunotherapy field. And now we want to expand this and sophisticate the system with a more optimal different extracellular vesicles, or also using the nanoparticles to deliver better LCOR to the tumors and do all the regulatory studies to make this a real therapy in the future. And with that, I want to thank you, the entire team, especially Ivan Perez, who is the PhD student, actually the first PhD student of the lab, and the PhD student that has led this project. This study was published like 1 month ago in Nature Cancer, so we are extremely excited about it, and he did an amazing and impressive job in the lab. And also all the collaborators, and Joan Albanese, who has been very implicated also in this study, and our funding agencies. And with that, I'll be very happy to have and attend your questions.
Thank you, Toni. Thank you, Toni, for the nice presentation. I will take the opportunity to congratulate you, but just to ask a couple of questions, like chairperson. Very general, one is not. So LCOR enhancing will work in the context of a beta-2 microglobulin mutation or a JAK2 mutation?
No.
Will not. No, you need the system more or less intact.
No, I mean, if there is any mutation in B2M, then we cannot overcome this because it goes through MHC1. So yeah.
And LCOR, I don't know, you showed me, maybe I missed. In breast cancer, LCOR expression is up, down. There is some alteration there.
So in luminal, well, in the normal mammary gland, LCOR is very high in the luminal compartment. As the neoplasia is happening, LCOR levels are decreasing, and in advanced stages, it's very low. Okay. But it acts as a tumor suppressor intrinsically, but also at the level of the immune system, as I showed you. But the levels are quite low, but still more expressed always. There is a differential expression within the tumors. It is more expressed in those cells that are more differentiated, but not with very high levels. You know.
And the last one, this is extremely general. So it was kind of a surprise that immunotherapy did not work so well in breast cancer. I know. So what are there many thoughts about this? Why it doesn't work so well? Like in breast cancer like in lung? What is the difference here? Is the mutation rate, or what is the difference?
So different things, and everything is sort of speculations or whatever is known in the field, is that breast cancer tumors in general are more cold, so they have less infiltration, especially ER positive. So ER positive is very cold tumor. And triple negatives is a little bit more of a mix. Has a bit more of genomic instability also, so there is a little bit more of infiltration, but still seems not to be enough to explain the lack of strong responses in triple negative breast cancer. That's why immunotherapy is only thought for the moment for triple negative breast cancer. Also, there are trials in ER positive. But right now, the most of efforts are focused on the triple negative, but still not with very strong responses and not with clear answers. And we try to, shed some light here about the heterogeneity, phenotypic heterogeneity that can be very important.
Okay. Thank you. Remember, those following online, they can ask questions using the chat.
First of all, congratulations for the nice talk. And I have couple questions. One is looks that LCOR is very good molecule and that play important roles in the cancer. But I wonder why you don't characterize who is the partner and how modification can alter in the different kind of condition that you have studied. And the other one is that looks really nice, the result with the exosomes, but you only have characterized on the mRNA level. But have you checked the peptide in the peptidomics in these exosomes? Because it's important roles in cancer and immunotherapy. And recently results in the Albert Head lab has shown that the peptidomic in exosomes is relevant for the immunotherapy. Thank you.
So the first question I think is related to the mechanism, right? Which partners can alter the gene expression in chromatin. We are currently doing this. Yeah, I mean, we focus more on the preclinical aspects, the function, how we can characterize this heterogeneity, and now we are going deeper in the mechanism. We have already some clues. We've done different assays, and we want to do more. And we have different candidates, chromatin remodeling candidates that can interact. Actually, we see that interact with LCOR, and these can be the ones that can mediate also this function.
Yeah, because in the literature, in the UniPrep, they know that it's around 20 proteins that are interacting with them, but can be changed the interactome depend of the condition that are around, and some histone also is well characterized that interact well with them and related with phosphoproteomics and some other kind of modification that can be there. Okay.
Yeah. I mean, and we are exploring also different conditions with and without interferon, and we see how the interactome is changing, and it's quite exciting, and we definitely are approaching this. The second question was about the exosomes. So the way we do the therapy is just basically we increase the expression of LCOR, and we see how the mRNA is selectively packed there. And then we compare with the other exosomes without LCOR, without the mRNA. So we have the control condition that there are exosomes, whatever peptidomics there, we don't know, but also in the LCOR condition, we have the peptidomic or immunopeptides. And the main difference is the mRNA of LCOR. So now we, of course, need to, as I said, sophisticate more these. These are a preliminary proof of concept that we added to this study to show that the LCOR therapy is feasible. And we have now projects to also to move forward on this direction.
Well, that was an amazing talk. Thank you very much. So I was wondering about, the first part where you describe how cancer stem cells have low interferon responsiveness. So two questions. One is, to what extent do you think that this is something that happens in other solid tumors? Have you looked at that or not? And second, so to what extent do you think that the invisibility that this gives to tumor cells, to cancer stem cells, is the only effect, beneficial effect, that low interferon has. So because, you know, high interferon also has other detrimental effects, high apoptosis, other things. So maybe have low interferon responsiveness has other beneficial effects.
Yeah, so the first part, I don't remember. I will I will answer the second, then you ask me again. But so you mean the intrinsic effects, right, of the interferon? Yeah, so we have done assays comparing like immunodeficient mice, completely immunodeficient, and immunocompetent, basically to compare this. And of course, LCOR in the tumors, when you increase LCOR and there is more interferon response in the immunodeficient mice, it also reduces the tumor growth. But it's a reduction. It's an important reduction, but it's not completely eradicating the tumors, not even close. The big difference is when we add immune system, but not only when we add immune system, when we add immune system plus immunotherapy. There we have, you know, a completely different magnitude of response. And yes, I mean, both things are important, but the effect of the immune system is totally, you know, outstanding, outcompeting the other. And the first part of the question was the interferon.
Other cancer stem cells.
Other cancer stem cells. Interferon, I mean, as I showed one slide, that in general is something that tumors try to avoid as they want to progress. This is based on all the literature also. Although interferon has so many pleiotropic effects, in some cases can be promoting, but this is due to other, you know, pro-inflammatory factors, whatever. But in breast cancer, there are 2 other papers showing that breast cancer stem cells have low interferon response. In other cancer types, right now I don't have anyone in mind, but I'm sure it's something general of other cancers. I'm sure, I don't know, but I guess. Yeah.
Hi, Toni. Thank you. It's a beautiful talk and beautiful work. I had just a very quick question. So in the model where you overexpress LCOR, so what do you have there? Cancer stem cells that become differentiated, or cancer stem cells that express the APM because you are overexpressing LCOR?
So the cancer stem cells get differentiated also, because in my previous study in the postdoc at Princeton, we basically were looking to that. We found LCOR because it was a factor that popped up when we induce differentiation and in the differentiation of the cancer stem cells. So it has these 2 effects. And this effect we showed that was mediated through the interferon response. But now what we found, and it was very striking, is that LCOR is not doing this indirectly. The antigen presentation is also directly orchestrating these genes of the antigen presentation.
The question would be, what's silence LCOR in the cancer stem cells?
So one of the things that I didn't explain it, but there is one slide of, again, my previous study. We found this microRNA 199 that directly represses LCOR. This microRNA is highly expressed in the cancer stem cells. It's part of the regulation of also epithelial to mesenchymal transition is involved, is downstream of TWIST1, and it can repress LCOR. We also are exploring even now more pathways that can regulate LCOR, because it's actually, we are lucky in that sense, or unlucky, that is not very well explored, these genes. So we have a lot of room to explore many things, and we are also exploring any kind of pathways that can regulate LCOR.
Yeah, thank you.
Hi. NK cells are the cells that are responsible to remove this kind of cells that express low levels of antigen-presenting molecules. What are there in the breast how are there in the breast cancer? And also these cells do not express PD-1, so they are going to be refractory to PD-1 therapy. So other therapies that are immunotherapies are expand them will be good for this kind of cells.
So yeah, that's a good point. NK cells detect low levels of HLAs, for example, and kill those cells. but we know that these cancer stem cells also express other suppressive ligands of NK cells. It's something that we have not explored, but is an explanation why NK cells, at least also in our assays, because we have checked that, are not killing these low MHC cells, cancer stem cells in this case. And the second part, you asked if there is a specific immunotherapy for NKs or?
No, yeah, that these cells do not express PD-1, so the data you show on PD-1 will not augment the NK cell function. As you say, there are other factors that will immune suppress these cells.
Yeah, I mean, all that I'm showing is through the T cell function, adaptive immune system. And in the in vivo assay, there was a condition that I didn't specify, that is with the depletion in vivo of the CD8 compartment. So when we deplete the CD8 compartment and we apply immunotherapy plus LCOR, the effect that we see is not there. So these tumors still progress. So showing how our combination is specific of the action of the T cells in this system. Yeah.
Okay, we'll take a couple of questions from the outside online attendees. One is, you explained that cancer stem cells, they have immunoevasion, but at the same time, they are more resistant to classical chemotherapy. So why is that? It's a very general question.
So they are immunoevasive because of the mechanism I showed, and it's known from many, many years that they have different mechanisms to avoid the classical immunotherapy due to they have pumps to take out the drugs inside, they have enzymes to metabolize the drugs, they have different oxidative stress, and also this is associated in many cases of population that are quiescent cancer stem cells. These cells are not quiescent, what I show you now. But also many mechanisms of chemotherapy resistance are due to their quiescence. So I'm not sure if I'm getting to the point of the question, but
it's a very wide question about the resistance of cancer stem cells. Yeah. Another question is, how far are these treatments for humans? And this can be related to mRNA therapy in cancer. How well developed is that compared with other therapies?
So I know there are, I've been checking now some companies, if they do it, there are some few companies that are starting, but there is no mRNA therapy yet in cancer. So it's still on development, and actually we want to try to move it there also in our case, because we have a patent that protects this. So we are now studying possibilities to move it to the patients.
Perfect. Thank you very much. Once again, congratulate the speaker. Thank you very much.
Thank you.